When we look out at like a perfectly manicured suburban lawn, there's this overwhelming expectation of just absolute control.
Oh, absolutely. It's essentially a green carpet we've installed.
Yeah,
Yeah.
exactly. I mean, we water it on a precise timer, we fertilize it with these, specific chemical ratios, and we mow it down to an exact millimeter height.
Right. And we expect it to be this completely sterile, static environment.
We really do. We want it to bend entirely to human will, sort of like an outdoor living room.
Well, it's a highly engineered landscape. Honestly, it's perhaps one of the most unnatural environments we interact with on a daily basis.
Yeah, that makes sense.
We construct these spaces to be botanical monocultures, and we aggressively remove pretty much anything that isn't the specific turf grass we've selected. We pour immense resources into fighting off nature's attempts to diversify that space.
But then, you zoom in. Like, if you get down on your hands and knees and actually look at the soil level, you realize that pristine green carpet is actually a battlefield.
It really is. It's deeply complex.
Yeah, it's this microscopic jungle where wild, ancient evolutionary warfare is just happening right under our feet. Millions of organisms are fighting for resources, adapting to the poisons we spray, and finding loopholes in our landscaping strategies.
And the organism winning that war, the one that has truly mastered the suburban environment, well, it isn't the grass.
No, not at all.
The turf grass is really just the stage setting. It's for the organisms that have figured out how to use our obsession with lawns to their absolute advantage. The hitchhikers and the opportunists, they're the true masters of suburbia.
And that brings us to the mission of today's deep dive. We're going on an exhaustive, microscopic and ecological exploration of a tiny organism you have almost certainly stepped on.
Probably cursed at, too, when it ruined the look of your yard.
Oh, yeah, definitely.
Yeah.
Likely ignored entirely otherwise. We're talking about Panaeolus Foenisecii.
Commonly known as the Brown Mottlegill, or, fittingly, the lawnmower's mushroom.
Right, the lawnmower's mushroom. And to understand just how incredibly sophisticated this organism is, we're pulling from a really wide range of sources for you today.
We've got over 2 centuries of taxonomic history, some modern chemical extraction profiles,
And
and
the one I'm most excited about.
yeah, the groundbreaking August 2025 genomic assembly paper.
Exactly. Published by the Institute of Microbiology at the Chinese Academy of Sciences.
That 2025 paper is huge. It fundamentally shifts how we view saprotrophic fungi in urban environments.
Because it gives us the code, right.
Right. It provides the actual molecular source code for how a seemingly fragile little brown mushroom manages to conquer such a hostile, heavily managed habitat.
Because this unassuming little mushroom has an incredibly complex story, today we're going to explore how it has entirely adapted its physical evolution to, well, literally hitch a ride on our landscaping equipment.
And how it completely abandons certain chemical pathways in favor of building an internal antioxidant armor to survive the sun.
Which is wild. And how its massive genetic toolbox turns it into a living, breathing biosensor for urban pollution. But, okay, let's unpack this. Before we get into heavy metal bioaccumulation and DNA sequencing, let's talk about what this thing actually looks like.
Because if you have a lawn or you go to a local park, you have probably seen this.
For sure. It's ubiquitous, right?
Ubiquitous across North America, Europe, and pretty much any temperate region. Anywhere humans have decided to plant and manage grass, this fungus has followed.
So physically, we are talking about a very small brown agaric.
An agaric just means, a classic mushroom with a cap and gills underneath.
Right. The cap is usually tiny, maybe like 1 to 4 centimeters in diameter. It starts out shaped like a little bell or steeply conical as it pushes up through the grass blades.
And then as it matures, it expands to a broader, more convex shape.
Sometimes it keeps a low, obtuse little bump right in the center, which I think is called an umbo.
Yes, an umbo. But the key physical trait, the word that mycologists use to describe it in every single field guide, is hygrophanous.
Hygrophanous. Okay, explain that, because it's super important.
It's a crucial diagnostic feature. And the physics of how a hygrophanous cap works are actually fascinating. It isn't simply changing color like a chameleon does.
It's not a pigment thing.
Exactly. The color change is entirely dependent on how much moisture the cellular structure of the mushroom is currently holding, because fungal tissue is incredibly porous.
Like a sponge.
Basically. So when the cap is fully saturated with water, say right after you run your sprinklers in the evening or after a heavy morning dew, the water completely fills the spaces between the hyphal cells.
So it actually changes how light reflects off the mushroom.
Yes, it alters the refractive index of the tissue. So when it's soaking wet, the cap appears as this rich, dark reddish brown, almost like a dark chocolate or chestnut color.
And the surface looks smooth and uniform.
Right, very matte. But as the morning sun comes out and the ambient humidity drops, the mushroom begins to lose that moisture, and it usually evaporates from the center of the cap outward.
And as that water leaves the cells, the color fades, which creates this really incredibly distinct two-toned look, right?
Exactly. As the center dries, air replaces the water between the cells, which changes the refraction again. So the center fades to a pale, creamy beige or a light tan color, while the outer margin of the cap might still hold moisture and remain dark brown.
That's so cool.
It creates a really striking banded or zoned appearance. Anyone trying to identify this mushroom has to realize that its color is not a static characteristic.
It's basically a real-time physical response to the immediate microclimate of your lawn.
Precisely.
Then we have the stem, or the stipe. It's slender, fragile, usually 4 to 10 centimeters long, kind of a bit paler than the cap, sometimes with a slightly powdery texture near the top.
And importantly, it has no ring on it.
Right, there's no skirt-like remnant of a partial veil that you see on some other mushrooms.
Yeah.
But the real giveaway, and the reason it gets the name Brown Mottlegill, is hiding underneath the cap. The gills.
Yeah, the gills. Underneath the gills are, well, in botanical terms, we say adnate to adnext.
Meaning they physically attach to the stem, right?
Yeah, exactly. They attach directly to the stipe, and they are packed quite closely together. Now, when the fruiting body is young, the gills are a pale, creamy cinnamon color. But as the reproductive cycle progresses, they don't darken evenly.
They get splotchy. They look almost, I don't know, pixelated or marbled.
They become mottled. And this visual effect is caused by the spore maturation process itself.
Because the spores are developing on these microscopic structures called basidia, right?
Yes, basidia, which line the faces of the gills. In Panaeolus Foenisecii, these basidia do not mature in unison.
Oh, so patches of spores are on completely different schedules.
Right. Patches on different parts of the gill surface reach maturity at completely different times.
Yeah.
And the fully mature spores are very dark, a dark purplish brown to almost black.
So you end up with a mosaic.
Exactly. Mosaic of light, immature patches, and dark, mature patches across the entire gill surface.
That mottled look is a huge clue if you're trying to identify it. But man, trying to figure out exactly what this mushroom is and where it belongs in the grand scheme of nature has been an absolute nightmare for scientists.
Oh, a total nightmare.
We're looking at a taxonomic turf war that lasted for over 2 centuries.
Taxonomy with little brown mushrooms is notoriously difficult. Macroscopic features, the traits you can see with the naked eye, are often convergent.
Meaning they end up looking the same even if they aren't related.
Right. Different, completely unrelated species will evolve similar shapes, colors, and structures simply because they're trying to survive in similar habitats. Form follows function.
Let's trace a timeline here because our sources show how much this poor fungus got bounced around. It was originally described way back in 1800 by the mycologist Christian Hendrik Persoon.
And he named it Agaricus Foenisecii.
Because back then, if it had gills and a stem, it was basically just tossed into the giant bucket of the genus Agaricus.
Pretty much. And the specific epithet Foenisecii translates roughly to of the hay harvest or haymaking.
Which tells you right from the start what ecological niche people associated it with. They were finding it in hayfields, in mowed pastures, in cut grass.
Right. But as microscopy became more advanced in the 19th and 20th centuries and taxonomy became more refined, that broad Agaricus bucket was broken apart.
And Fungal went into, like, the witness protection program of mycology. I mean, it was moved into the family Bulbiciaceae. Then some authorities insisted it belonged in Strophareaceae. Others argued for Satharellacea.
It had so many names: Panaeolus Foenisecii, psilocybin, Foenisecii, Sathyra Foenisecii, Satharella Foenisecii.
It's like every time someone looked at it under a better microscope, they changed its name.
Well, the primary source of all this taxonomic chaos came down to the spores themselves.
Right.
See, the genus Panaeolus, which includes species like Panaeolus cincellus or Panaeolus papilionaceus, is defined classically by having spores that are completely smooth and jet black.
But when mycologists put Foenisecii under a high-powered lens, they saw an anomaly.
A glaring anomaly. The spores are dark brown, not true black. And more importantly, they are not smooth at all.
They're warty.
Yes, the technical term is verrucose. They have this minutely roughened, bumpy texture all over the surface.
So because of that distinct microscopic difference, many 20th century taxonomists drew a hard line, right?
They did. They decided that a rough brown spore could not possibly belong in a genus defined by smooth black spores. So they created an entirely separate genus just to hold this oddball called Panaeolina.
Panaeolina, just for this one mushroom.
Basically, for decades it was classified as Panaeolina Foenisecii, sitting essentially alone as the type species of this tiny, isolated genus.
But then we hit the genetic revolution. We stopped trying to guess family trees based on what a spore looks like under a microscope, and we started reading the actual DNA.
And the modern molecular phylogenetics completely rewrote the story.
Which is wild. How exactly did they do that?
Researchers utilized multi-locus phylogenetic studies to settle the debate. Rather than relying on a single gene, which can sometimes be misleading, they sequenced multiple independent regions of the genome.
Like checking multiple forms of ID.
Exactly. They looked at the nuclear internal transcribed spacer, the ITS region, which is essentially the universal barcode for fungi.
Okay.
They sequenced the D1, D2 domains of the large subunit 28S ribosomal DNA. They examined protein-coding genes like the elongation factor 1-alpha, TEF1-alpha, and the RNA polymerase II second largest subunit, RPP2.
That is just a staggering amount of genetic data. They threw the entire molecular toolkit at this mushroom to definitively map its ancestry.
And the data returned an unequivocal result. The genetic sequences prove that this mushroom is nested deeply and directly inside the Panaeolus genus.
Wow.
It isn't a distant cousin, and it isn't a sister lineage that branched off early on. It is a core member of the Panaeolus family tree.
Okay, wait, I have to stop you there. Because if modern sequencing proves it belongs in Panaeolus, a family famous for having perfectly smooth spores, why does this one specific mushroom have rough, warty spores?
It's a great question.
Isn't that an evolutionary regression? It feels like it suddenly mutated a complicated texture for no reason.
It isn't a regression at all. It's actually a highly specialized adaptation driven entirely by its habitat.
Okay, how so?
To understand why those spores became warty, we have to look at the broader ecology of the Panaeolus genus. The vast majority of species in this genus are obligate coprophiles.
Meaning they love dung.
Exactly. They are evolutionary specialists adapted to live on and digest the dung of large herbivores. They thrive in pastures, breaking down the highly processed, nitrogen-rich waste of cows, horses, and sheep.
Okay, so a normal Panaeolus sits in a field, waits for a cow to drop a patty, and then sets up shop.
Right.
But Panaeolus Foenisecii lives on my front lawn. There are no cows wandering through the suburbs. It's like the fungus realized that waiting for roaming livestock was an outdated, unreliable business model, so it completely hijacked human suburbia instead.
That's a great way to put it. It executed a massive ecological shift. It moved from being a coprophile, a dung liver, to a saprotroph, specifically a litter decomposer.
Okay.
Its underground mycelial network, adapted to feed on decaying plant matter, dead grass roots, and the dense layer of satch that constantly builds up in human-managed lawns. This is a brilliant example of anthropogenic mutualism, a relationship shaped entirely by human activity.
We basically built the perfect machine to feed and spread this mushroom, and we had no idea we were doing it.
A lawnmower.
The lawnmower. It's the ultimate irony. A homeowner spends thousands of dollars on fertilizer, aerators, and a brand new riding mower to achieve a pristine lawn, and the mower itself is the exact vehicle the fungus uses to conquer the yard.
The lawnmower provides two critical services for the fungus. First, by constantly cutting the grass, the mower generates a continuous, unending supply of finely shredded grass clippings.
Which is food.
Massive amounts of food. It provides an easily accessible source of cellulose. We are essentially pre-chewing the mushroom's food, dropping it directly onto the soil surface where the mycelium can rapidly colonize it.
And the second service is dispersal, which is where your warty spores come back into play.
Precisely. Let's think about the physics of a lawnmower blade spinning at thousands of revolutions per minute.
It's basically a blender.
Right. When that blade sweeps over the lawn and strikes a patch of mature Panaeolus Foenisecii fruiting bodies, it doesn't just cut them, it obliterates them.
It creates a violent aerodynamic draft.
Yes, a draft that flings millions of spores across the yard. Now, if those spores were smooth, like its ancestral cousins, they would face a huge problem in a suburban environment.
Because a smooth spore on a smooth blade of turf grass is just going to slide right off. The next time the sprinklers turn on, or it rains, a smooth spore just washes away. It gets pushed deep down into the mineral soil, far below that nutrient-rich thatch layer, or it simply blows completely off the lawn and onto the hot concrete of the driveway where it'll just die.
But the warty varicose spores of P. Foenisecii completely change the physical dynamics. That minutely roughened texture acts like microscopic Velcro.
Microscopic Velcro, I love that.
It creates immense physical friction. When the spores are flung into the air, they catch and cling tightly to the jagged edges of torn grass blades, to tiny soil particles, and even to the metal of the lawnmower blade itself.
So the mower literally picks them up, carries them to the other side of the yard, and the friction ensures they stick exactly where they need to be.
Yes, suspended in that humid, nutrient-rich thatch layer, perfectly positioned to germinate.
It literally evolved a new microscopic anatomy just to ride our landscaping equipment.
It is a stunning example of evolutionary adaptation occurring in real time alongside human residential practices. And yet, what is perhaps even more fascinating is that despite this extreme level of specialization for suburban lawns, it hasn't completely discarded its ancient genetic roots.
Oh, you're talking about the 2015 study from Sri Lanka.
Yes.
This blew my mind when I was reading the source stack. Researchers were surveying dry zone forest reserves in Sri Lanka environments, completely isolated from manicured lawns, sprinklers, or, John Deere mowers.
Right.
And they found Panaeolus Foenisecii growing directly out of wild elephant dung.
And they confirmed the identification using the ITS and 28S ribosomal DNA sequencing we discussed earlier, so there was no morphological confusion.
It was definitively the lawnmower's mushroom.
Definitively. What this proves is that the organism retains a remarkable degree of ecophysiological plasticity.
Meaning it can switch back if it needs to.
Exactly. Its metabolic toolkit, the enzymes it produces to digest food, is still perfectly capable of breaking down the high-nitrogen, fibrous plant matter found in herbivore dung when it's available in the wild.
It hasn't forgotten how to be a coprophile. It just prefers the easy, consistent, heavily irrigated buffet of our lawns.
It's the ultimate opportunist.
But, living on a suburban lawn isn't exactly a vacation. It's actually a brutally harsh environment.
It really is.
If you're a tiny, fragile mushroom, you are fully exposed to the sun, you get baked on hot summer afternoons, and you are subjected to wild, rapid swings in humidity. Soaked by automatic sprinklers at dawn and bone dry by noon.
Surviving that requires a very specific, heavy-duty internal chemistry.
Which leads us to the biochemistry of this thing, which is arguably its most impressive survival mechanism. And it involves a fascinating evolutionary pivot regarding the chemicals it chooses to produce.
Yes, it does.
Let's address the hallucinogenic elephant in the room. The genus Panaeolus is famous for producing highly psychoactive tryptamines.
Right. Species like Panaeolus cyanescens, often called the blue meanie, or Panaeolus cinctulus, the banded mottlegill, synthesize significant amounts of psilocybin and psilocin.
Because our lawnmower mushroom is genetically nested right in the middle of this family and looks very similar, it was plagued by chemical misinformation for decades.
Oh, absolutely. If you look at older mycological field guides published in the 1960s or 70s, you will frequently see P. Foenisecii erroneously listed as a hallucinogenic mushroom.
People assumed that because of its lineage, it must contain the same compounds.
Exactly.
Our sources note that amateur foragers used to accidentally harvest it from their lawns, thinking they had found a psychedelic mushroom, only to ingest it and be entirely disappointed.
Which is dangerous, but yes.
So, what is the actual chemical reality? We need to be clear and look strictly at the analytical data here.
Modern analytical chemistry has definitively settled this debate. Researchers have utilized high-sensitivity liquid chromatography coupled with mass spectrometry.
Okay, break that down for us. What does that actually do?
It's an incredibly precise technique that physically separates the molecules within a sample and measures their exact molecular mass, allowing us to identify compounds down to trace amounts.
Oh, wow. Okay.
When extensive collections of P. Foenisecii are run through this equipment, the results consistently show that it contains either non-detectable or completely negligible trace amounts of psilocybin and psilocin.
Trace amounts meaning it is far, far below any active threshold. It would have absolutely no physiological or psychological effect on a human being.
None whatsoever. Furthermore, it completely lacks the characteristic chemical reactions seen in psychoactive Panaeolus species.
The bruising.
Right, it does not bruise blue when the flesh is damaged.
Because in a species like P. cyanescens, when the cellular tissue is broken, enzymes rapidly oxidize the psilocin upon exposure to oxygen, right?
Yes, creating a distinct blue pigment. But because P. Foenisecii lacks meaningful amounts of psilocin, it simply cannot produce that blue bruising reaction.
So it's a Panaeolus, but it completely abandoned the psychedelic pathway. But wait, I have to challenge this logic.
Go ahead.
Evolution is fundamentally driven by efficiency and survival advantage. If the complex genetic machinery to produce psilocybin was already present in its evolutionary ancestors, which we know it was, why on earth would it turn off those genes?
That's the big question.
Psilocybin is thought to deter certain insects from eating the fungus. So giving it up is like disarming yourself in a war zone. Why make that trade-off?
You are correct that secondary metabolites like psilocybin serve an ecological purpose, likely as a feeding deterrent. But biosynthesizing complex molecules is incredibly energy-intensive for a fungus.
It takes a lot of work to build those molecules.
Exactly. It requires a cascade of specific enzymatic reactions, all of which burn cellular resources. For a coprophilous mushroom living on a stable, nutrient-dense pile of dung, perhaps that energy expenditure is justified by keeping dung beetles or fly larvae at bay.
Makes sense.
But for a saprotroph living on a highly exposed suburban lawn, the primary existential threat is not necessarily a bug. The primary threat is the environment itself.
The sun,
Yeah.
the heat, the rapid drying.
Precisely. On a manicured lawn, the delicate, thin-walled cellular tissue of the mushroom faces brutal, direct ultraviolet radiation from the sun.
And UV radiation is destructive.
Very. When UV radiation hits organic tissue, it creates reactive oxygen species-free radicals.
Ah, free radical.
These are highly unstable molecules that aggressively strip electrons from other molecules, causing massive cellular damage, destroying DNA, and accelerating cell death. This is extreme oxidative stress.
So if you're a mushroom sitting on a lawn at 2 p.m. in July, you need serious chemical protection.
You do.
And this is where the biochemical pivot happens. A seminal 1963 study by researchers Tyler and Smith looked into what the mushroom was actually producing since it wasn't psilocybin, and they found it was hoarding an entirely different set of primary indole derivatives.
They discovered that Foenisecii synthesizes massive amounts of 5-hydroxytryptophan, which we know as 5-HTP.
Wow.
It also produces high concentrations of serotonin, known scientifically as 5-hydroxytryptamine, and it produces 5-hydroxyindoleacetic acid, or 5-HIAA.
Okay, to anyone who has looked at the supplement aisle of a pharmacy, those are incredibly famous neurochemicals.
They certainly are.
5-HTP is the direct biochemical precursor your body uses to make serotonin, and serotonin regulates mood, sleep, and digestion in humans. But why is a mushroom manufacturing a mood stabilizer?
Well, in the fungal kingdom, and indeed in many plants, serotonin and its related compounds like melatonin serve a very different primary function. They act as exceptionally potent antioxidants.
Oh, they're chemical scavengers.
Exactly.
So when the UV rays hit the mushroom and create those destructive free radicals, the high concentrations of serotonin essentially sacrifice themselves to neutralize the threat.
Yes, they donate electrons to the free radicals, stabilizing them before they can tear apart the fungal DNA. It's an internal chemical sunscreen.
It's a brilliant, highly evolved internal armor. By dedicating its enzymatic pathways, specifically upregulating enzymes like tryptophan hydroxylase and aromatic L-amino acid decarboxylase, the mushroom converts its available L-tryptophan into serotonin rather than psilocybin.
It prioritizes surviving the harsh, exposed microclimate of the lawn over producing complex psychoactive deterrents.
But doesn't it still get eaten by bugs if it gives up the psilocybin?
That is the secondary genius of this adaptation. High levels of exogenous serotonin actually act as a powerful natural feeding deterrent against common lawn pests, like certain soil nematodes and insects.
Oh, wait, really?
Yes. A massive dose of indoles overwhelms their digestive or nervous systems. So by pivoting to serotonin, the mushroom achieves a dual-purpose defense: maximum UV protection and pest deterrence, optimized entirely for the lawn environment.
It's just a masterclass in resource allocation.
Oh.
And speaking of chemical defenses, our sources highlighted another wild trick this mushroom has up its sleeve, specifically protecting those warty spores we talked about earlier.
Yes, the spores are heavily chemically armored.
How armored are we talking?
Well, in laboratory settings, mycologists use chemical reagents to test the resilience and composition of fungal tissues. If you take the spores of P. Foenisecii and apply a drop of concentrated sulfuric acid H2SO4, you would expect a violent reaction.
Because concentrated sulfuric acid is a powerful dehydrating agent, right? It rapidly strips water from organic molecules.
Exactly, usually charring them black and dissolving them in seconds.
But when you drop it on these spores, nothing happens. They just sit there.
They do not discolor and they do not dissolve. They remain completely structurally intact.
That's insane.
This extreme chemical resistance is a specialized trait unique to the subfamily Panaeolus. This heavy armor likely evolved to help the spores survive the highly acidic digestive tracts of grazing animals in their ancestral coprophilous days.
But on a modern suburban lawn, that same chemical resilience allows the spores to survive the harsh synthetic fertilizers, weed killers, and soil amendments that homeowners constantly dump onto the grass.
Right, it's pre-adapted to human chemicals, and there's one more chemical trick that is really useful for field identification, the KOH test.
Potassium hydroxide.
Yes, KOH is a common chemical reagent used by mycologists. In many mushroom species, applying a weak solution of KOH to the dry surface of the cap causes a dramatic, immediate color change.
Like turning bright red or yellow.
Or instantly black, yeah. This happens due to the specific pigments in the cap reacting with the strong base. But when you apply KOH to the cap of P. Foenisecii, it yields a completely negative or dull, watery gray reaction.
A very simple but effective piece of forensic evidence to verify you are looking at the lawnmower's mushroom.
Exactly.
Okay, so we've looked at the warty spores that let it surf lawnmower blades, and we've explored the internal serotonin factory that acts as its chemical sunscreen. To truly understand how a tiny organism manages to engineer all of these complex systems, how it builds the enzymes to eat tough grass and synthesize massive amounts of antioxidants, we have to look at its actual source code.
We are transitioning from the phenotype, the physical and chemical traits we can observe, to the genotype, the underlying genetic blueprint that builds those traits.
And the data we have here is absolutely cutting edge.
It really is.
According to our source SEC, in August 2025, a massive milestone was reached. The Institute of Microbiology at the Chinese Academy of Sciences, the CAS, published the very first high-quality haploid reference genome assembly for Panaeolus Foenisecii.
Specifically, they sequenced strain 22TS01220.
This represents a monumental leap forward for fungal genomics, doesn't it?
Oh, massive. Previously, as we discussed, we only had fragments of its DNA, the ITS and 28S markers.
Which are essentially just genetic barcodes. They tell you what the species is, but nothing about how it works.
Right. A reference genome is entirely different. It is the complete, unabridged instruction manual for the entire organism.
I really want to dig into the technical specs of how they map this because the technology is fascinating. Our sources note it was generated using PacBioSequel technology and assembled via the Fly algorithm.
Yes.
Let's translate that from geneticist speak. How does this actually work compared to older methods?
Okay, so traditional DNA sequencing, like the older Illumina platforms, relied on short-read technology. It would chop the DNA up into tiny fragments, read segments that were maybe 150 to 300 base pairs long, and then software would try to piece them back together.
I always think of that like trying to put together a 10,000-piece jigsaw puzzle of a clear blue sky.
That's a perfect analogy.
Because the pieces are so small,
Right.
and so many of them look identical, especially with repeating DNA sequences, it's incredibly difficult to know exactly where they overlap. You end up with a lot of gaps and errors.
Exactly. But PacBioSequel is a long-read sequencing technology.
So the pieces are bigger.
Much bigger. Instead of reading a few hundred base pairs, it can read continuous, unbroken strands of DNA that are tens of thousands of base pairs long. It provides massive, already assembled chunks of the puzzle.
And the Fly algorithm.
The Fly algorithm is the specialized mathematical software designed specifically to handle these massive long reads. It uses complex graph theory, particularly De Bruijn graphs, to map the overlaps between these giant DNA fragments, easily resolving the repetitive sequences that confused older software.
It stitches these long reads together into continuous genomic maps.
Exactly. And because of this advanced technological pairing, the resulting genome assembly for the mushroom is incredibly continuous and robust.
The paper states it spans a total sequence length of 51.8 megabases. That's 51.8 million base pairs of DNA. And the software consolidated all of that data into just 22 contigs.
Now, a contig is a contiguous, unbroken, overlapping sequence of DNA. To have an entire 51.8 million base pair genome represented in just 22 unbroken stretches means the researchers achieved a near-perfect map.
They have virtually no gaps in the data.
Practically none. The scaffold N50, which is a standard metric used to judge the quality of a genome assembly, is 4 megabases.
Meaning that at least half of the entire genome is contained in massive, continuous stretches of DNA that are 4 million base pairs or longer.
Exactly right.
The researchers also noted it is a chromosome-free contide-level reference. Does that mean it doesn't have chromosomes?
No, it certainly has physical chromosomes inside its nucleus. That phrasing simply means that while the data is highly continuous, the researchers haven't yet mapped exactly which of those 22 contigs stitched together end to end to form the final physical structures of the chromosomes.
Got it.
But for the purpose of identifying functional genes, metabolic pathways, and evolutionary traits, a contig-level assembly of this pristine quality is an absolute goldmine.
Let's talk about that total size. 51.8 megabases. Is 51.8 million base pairs a lot for a mushroom?
It is massively expanded compared to typical saprotrophic agarics. Many related mushrooms have genomes that hover around the 35 to 40 megabase range.
Oh, wow.
When you see a genome balloon up to nearly 52 megabases, it tells you something profound about the organism's lifestyle. It tells you that the fungus is hoarding genetic tools.
It has a massive metabolic toolbox. What exactly is taking up all that extra space in the genome?
A significant portion of that extra genomic real estate accommodates a huge, heavily diversified arsenal of enzymes.
C-enzymes, carbohydrate-active enzymes.
Correct. We have to remember what this mushroom eats. Grass is incredibly tough.
Yeah, it's very fibrous.
It is structural plant matter built from cellulose, hemicellulose, and lignin. These are complex, stubborn polysaccharides designed by nature specifically to resist breaking down.
Right.
To decompose that thatch layer and extract the trapped nutrients, the mushroom needs a highly specialized array of digestive enzymes acting as microscopic scissors to cut those complex sugar chains apart.
And the expanded genome provides the immense genetic coding required to manufacture all these different enzymes.
Exactly, giving P. Foenisecii the metabolic flexibility to eat whatever kind of dead roots or grass clippings the lawnmower drops on it.
So it's heavily armed for digestion. What else is in the 51.8 million base pairs?
The genome also houses an expanded array of secondary metabolite BGCs, or biosynthetic gene clusters.
Okay, what do those do?
These are essentially clustered genetic assembly lines responsible for producing complex chemicals. This is the exact genetic machinery that builds the massive amounts of serotonin, 5-HTP, and protective antioxidants we discussed earlier.
This is where I want to bring it back to the real world, to you, the listener. It is incredible that this mushroom has evolved a massive genome to eat grass and synthesize serotonin to survive the sun. But how does mapping its DNA in a lab in 2025 actually impact human lives?
The biotech implications are profound and highly lucrative.
Really? How so?
Well, having this complete high-quality reference genome allows researchers to isolate the exact specific genes encoding the tryptophan decarboxylase and hydroxylase enzymes.
The super efficient biochemical factory that the mushroom uses to pump out serotonin.
Precisely. Once you know the code for the factory, you can copy it.
That's amazing.
Once you have the exact genetic sequence, you can synthesize those genes and insert them into plasmids. You can then introduce those plasmids into industrial microorganisms like specific strains of yeast or bacteria that are easy to grow in massive fermentation vats.
This opens the door for bioengineering pharmaceutical-grade 5-HTP and serotonin.
Exactly. Currently, synthesizing these complex neurochemicals chemically is expensive and difficult, but pharmaceutical companies could soon use the metabolic pathways perfected by a common yard mushroom to produce vital raw materials for antidepressants, sleep aids, and neurological medications at a massive, sustainable industrial scale.
The annoying little brown mushroom that pops up in the grass after you run the sprinklers could hold the genetic key to manufacturing the next generation of psychiatric medication. That is just incredible.
It perfectly illustrates why preserving and deeply understanding biodiversity is so critical, even in highly anthropogenic, human-made environments like suburban lawns. You never know where the next major biochemical breakthrough is hiding.
Okay, so on paper, in the genomic databases, this mushroom is an absolute marvel of evolutionary adaptation. But the same mechanisms that make it a biotech superstar actually make it a potential hazard on the lawn where kids are playing and dogs are running around.
That's true.
Because its superpower of digestion and absorption has a dark side.
We established that its genome is packed with enzymes, creating an extensive, shallow, highly aggressive mycelial network designed to rapidly absorb nutrients from the grass thatch.
But that underground network isn't picky. It acts like a highly sensitive biological sponge.
Very sensitive.
And because P. Foenisecii lives almost exclusively in urban and suburban soils, it is absorbing whatever we have spent the last 100 years putting into that soil.
It is an incredibly efficient heavy metal bioaccumulator. Our sources detail how the mycelium of P. Foenisecii actively concentrates specific toxic elements from its immediate environment, pulling them out of the soil and storing them in the fruiting body. the mushroom itself.
It specifically pulls in high levels of lead, cadmium, copper, and zinc. Where is it getting lead and cadmium on a random suburban lawn in the middle of a neighborhood?
The legacy of human industry. Decades of using leaded gasoline before it was banned have left the topsoil of roadside verges, parkways, and older urban lawns heavily contaminated with residual particulate lead.
Wow, even decades later.
Yes. And cadmium and copper are ubiquitous in modern agriculture and landscaping. They are frequently found as trace contaminants in synthetic lawn fertilizers, and they are active ingredients in certain types of fungicides, pesticides, and herbicides that homeowners routinely spray to achieve a perfect green lawn.
So the mushroom is just acting as a vacuum, sucking up all the chemical fallout of modern landscaping and urban traffic, concentrating it into a tiny brown package.
Because of this trait, environmental scientists actually utilize P. Foenisecii as a living bioindicator.
That's clever.
Rather than taking hundreds of expensive soil samples, researchers can simply harvest these common mushrooms from parks and lawns across a city. By measuring the heavy metal content concentrated in the fungal tissues, they can accurately gauge the ambient pollution levels of the surrounding urban environment.
The mushroom silently measures and records the toxicity of our cities.
Exactly.
But this creates a very real, practical safety issue.
Yeah.
Because this mushroom is literally everywhere. It pops up in the grass worldwide. And kids and pets, naturally curious, constantly put things they find in the grass into their mouths.
Which is exactly why P. Foenisecii is consistently one of the most frequently ingested mushrooms reported to poison control centers every single year. Parents see a toddler eating a brown mushroom from the yard and immediately panic.
But here is a critical distinction that our sources make, and we need to be very clear about the toxicology here. The fungal tissue of Panaeolus foenisecii itself is not inherently acutely toxic.
That is correct. It does not produce deadly cellular poisons like the amatoxins found in the death cap mushroom, nor does it produce severe neurotoxins. It is not trying to kill anything.
Yet, people, especially small children, frequently get sick when they eat it. The medical reports show mild to moderate gastrointestinal distress, nausea, stomach cramps, vomiting, diarrhea.
Yeah.
If the mushroom isn't making a poison, what is causing the illness?
The illness is largely attributed to the environmental pollutants the mushroom has absorbed.
Oh, wow.
When a child eats a handful of P. Foenisecii off a chemically treated lawn, they aren't just digesting fungal tissue. They are receiving a concentrated, bioaccumulated dose of the heavy metals, the residual weed killers, the pesticides, and the synthetic fertilizers that the mushroom's mycelium has spent weeks sponging up from the toxic topsoil.
That is terrifying.
The gastrointestinal distress is very often a direct physiological reaction to the urban pollution itself, simply delivered via the mushroom.
The mushroom isn't the poison. The pristine, chemically maintained lawn is the poison. The mushroom is just the messenger. That is wildly unsettling when you think about how we treat our yards.
It really is.
But beyond the heavy metals and fertilizer, there is another massive danger associated with encountering this mushroom, and that is the danger of misidentification.
Misidentification is perhaps the most severe risk in amateur mycology, particularly within the category foragers refer to as LBMs, or little brown mushrooms.
Because to the untrained eye, 100 different species of little brown mushrooms look completely identical.
Exactly.
And P. Foenisecii shares its exact habitat-manicured, irrigated, sunlit lawns with some much more problematic species. It grows side by side with toxic lookalikes like Conocybia pala and its psychoactive cousin, Panaeolus cinchillus.
This is where forensic differentiation becomes absolutely vital. If someone ingests a mushroom from a lawn, medical professionals and toxicologists need to know exactly what was consumed to determine the proper course of treatment.
Let's look at how you definitively tell the lawnmower's mushroom apart from the psychoactive Panaeolus cinchillus. And just to reiterate, we are simply reporting the chemical reality here based on the sources.
Right.
P. cinchillus contains psilocin, and P. Foenisecii does not. So how do you tell them apart physically if they are growing in the exact same patch of grass?
While both species have that water-reactive hygrophanous cap and share the identical habitat, there are clear markers. Macroscopically, P. cinchillus is generally a slightly stockier, more robust mushroom. The stipe is thicker.
Okay, stockier stem.
But the primary visual tell, the immediate forensic evidence, is bruising. Because P. cinchillus contains active psilocin, when the base of its stem is handled, crushed, or damaged, Oxygen interacts with the enzymes in the broken cells. This rapidly oxidizes the psilocin, causing the tissue to bruise a distinct blue or deep blue-green color.
And as we established when discussing its chemical sunscreen, Foenisecii lacks psilocin, so it will never ever bruise blue no matter how much you crush it.
Correct. The lack of an oxidation reaction is a strong indicator. The second definitive forensic tool is the spore print. This is how you confirm identity beyond a shadow of a doubt. If you remove the cap of the mushroom, place the gills face down on a piece of white paper, cover it with a glass, and let it sit overnight, it will drop millions of spores, creating a colored print.
Like a fingerprint.
Basically, P. cinctulus will drop spores that create a jet black print. P. Foenisecii will leave a dark brown or purplish-brown print.
It sounds subtle, but when you see them side by side on white paper, the difference between pure black and purplish-brown is very clear.
It is. And finally, if a toxicologist has access to a microscope, they look at the architecture of the spores themselves. As we discussed regarding its evolution, the psychoactive P. cinctulus will have completely smooth, featureless spores,
while P. foenisecii will display those distinctly roughened, warty varicose spores that it evolved to stick to the lawnmower blade. So if you are looking at a mushroom in your grass, the forensic toolkit to identify the lawnmower's mushroom is no blue bruising on the stem, a purplish-brown spore print instead of a pure black one, and if you have a microscope, warty spores instead of smooth ones.
It requires careful, methodical observation, but those biological markers are absolutely definitive.
It really is an incredible journey when you step back and look at the whole picture. We started out looking at this annoying, fragile little brown nuisance popping up in the grass, ruining the look of a perfectly manicured yard. But when you unpack the biology, Panaeolus Foenisecii is an absolute master of anthropogenic mutualism.
It is a survivor of the highest order. It recognized a massive, unexploited ecological niche, the American lawn, and completely restructured its biology to dominate it.
It completely altered its physical dispersal strategy,
Yep.
evolving microscopic Velcro spores specifically to catch a ride on the metal blades of our lawnmowers. It completely overhauled its internal chemistry, giving up an energy-intensive psychedelic pathway in exchange for an internal, high serotonin antioxidant armor to survive the brutal, unshaded sun exposure of suburbia.
It expanded its entire genetic architecture, ballooning its genome to nearly 52 million base pairs just so it could carry the massive toolbox of enzymes required to digest the tough grass clippings we constantly provide for it.
And while it does all of this, while it perfectly integrates itself into our landscaping routines, it sits there silently absorbing the chemical fallout of our cities, acting as a living biosensor for heavy metal pollution. It is a biological marvel hiding in plain sight right at the tips of our shoes.
It certainly makes you look at the ground differently, and it leaves us with a final provocative thought to ponder.
Oh, absolutely.
We spend immense amounts of time, money, and chemical energy curating our lawns. We view these manicured, heavily managed suburban spaces as sterile, artificial environment spaces that we have tamed that are somehow devoid of true, wild, adapting nature. But if Panaeolus Foenisecii has so rapidly and radically altered its genome, its chemical output, and its physical anatomy to perfectly exploit our lawnmowers, our chemical fertilizers, and our sprinklers, what other invisible evolutionary leaps are happening right under our feet in the very habitats we think we completely control?
The pristine green carpet isn't a static monoculture at all. It's a high-speed evolutionary proving ground, and the hitchhikers are adapting faster than we are. Thank you so much for joining us on this deep dive into the source material. We will see you next time.